Hi, Dr. Jeffrey Mark here. This week, I would like to answer some questions people have asked me about the immune system, the two approved messenger RNA vaccines put out by Pfizer, BioNTech, and Moderna, and immune resiliency. If you need a refresher on how the details on these vaccines and how they work, I would refer you to the previous two blogs on the website regarding these vaccines. Let me first start with the question about how long immune memory might last. The three primary aspects of immune memory are antibodies, killer T cells, and helper T cells. The following graphics and images are from the article in Cell, volume 181, issue 7, pages 1489 to 1501. The title of the article is Targets of T Cell Responses in SARS CoV2 Coronavirus in Humans with COVID-19 Disease and Unexposed Individuals. Circulating SARS CoV2 specific CD8 positive and CD4 positive T cells were seen in approximately 70% and 100% of COVID-19 recovered patients, respectively. CD4 positive T cell responses to spike, the main target of the present and current vaccines that are approved, were robust and correlated with the anti-SARS CoV2 immunoglobulin G and immunoglobulin A titers. The M spike and N proteins each accounted for 11% to 27% of the total CD4 positive response, with additional responses commonly targeting NSP3, NSP4, ORF3A, and ORF8, among other sites. For CD8 positive T cells, spike and M proteins were recognized, with at least 8 SARS-CoV-2 ORFs targeted. Of note, it was also found that SARS-CoV-2 reactive CD4 positive T cells in approximately 40% to 60% of unexposed individuals, suggesting cross-reactivity to T cell recognition between circulating common cold type coronaviruses and the SARS-CoV-2 coronavirus. There is a recently released paper, November 1st, 2020, that's entitled Immunological Memory to SARS-CoV-2 Assessed for Greater than 6 months After Infection. The authors included lead author Jennifer M. Dan, and they analyzed multiple compartments of circulating immune memory to SARS-CoV-2 in 185 COVID-19 cases, including 41 cases at greater than 6 months post-infection. They found that the immunoglobulin G antibody to the SARS protein was relatively stable over 6 months. Spike-specific memory B cells were more abundant at 6 months than they were at 1 month. The SARS-CoV-2 specific CD4 positive T cells and CD8 positive T cells declined with a half-life of 3 to 5 months. In their examination of antibody memory B cell, CD4 positive T cells, and CD8 positive T cell memory in SARS-CoV-2 in an integrated manner, they observed that each component of the SARS-CoV-2 immune system memory exhibited continued activity for at least 6 months. There have been questions about mutations in the SARS-CoV-2 spike protein in the UK. News outlets implied that new strains were developing, but most virologists believe these are variants to SARS-CoV-2 at this time, and it's hard to prove that they are more virulent or accounting for higher rates of infection in the UK. Reports of mutations were also brought up recently with the extermination of most of the population of mink in Denmark. People infected minks, which apparently have similar susceptibility of the SARS-CoV-2 virus, and then a mutation occurred where at least 12 humans apparently were infected back by the minks. There was concern that the spike or other proteins were changed enough that the current vaccines in development potentially would not be as effective or be ineffective. Most virologists believe that the spike protein remains stable enough for now so that there would be continued immune protection from the current two vaccines. Of course, coronaviruses like the flu can have significant seasonal variations and are always mutating, so we will have to see over time if this SARS-CoV-2 will behave more like other coronaviruses. As you may know, we get new yearly flu vaccines based on the most prevalent new variation of the flu virus, and our immunity starts to wane after a few months. We were initially worried that antibody counts initially dropped to undetectable levels after 3 to 4 months with the SARS-CoV-2, but with the study above that I just mentioned, other components of the immune system, like the memory T cells and helper T cells and B cells, may be involved in immune memory for at least 6 months. How long does the RNA last? Typically, the messenger RNA is made from a DNA template and can last several hours to about 1 day. After vaccination with the messenger RNA-based vaccine, the lipid bilayer surrounding the messenger RNA gets incorporated into muscle cells or lymphoid tissue at the injection site, and the cell will read the RNA instructions several times and will start making SARS-CoV-2 spike protein for several hours, up to 2 days, as the messenger RNA was specifically engineered to last at least 1 to 2 days. But then it will get broken down like any other normal messenger RNA. There have been questions about whether an enzyme called reverse transcriptase can convert the viral messenger RNA into DNA and then get incorporated into our human DNA. In theory, this might be possible, but it has not been demonstrated in any study with the current engineered RNA, messenger RNA that I know of. And this messenger RNA is in the cytosol and not in the nucleus of the cell, which houses our genetic library or DNA. There are questions of whether there are any additives to the current messenger RNA vaccines. From what I've heard from the companies, there are no preservatives or other additives other than saline and perhaps buffering agents to adjust for pH and other characteristics to keep the lipids in adequate suspension. Should people who have already had COVID-19 be vaccinated? The current CDC and FDA recommendation is that even people with whom have had COVID-19 be vaccinated, and the rationale may be that the immune responses may vary as people that have less severe symptoms may not have as vigorous an immune memory and lasting protection as people with more severe symptoms. The paper we mentioned seems to indicate immune memory is present for at least 6 months, but we don't know if the body were challenged with another infection with the same virus or a variation of the virus, if the body would mount an adequate protective immune response. We unfortunately do not know how long the vaccine protection would last as well. So the strategy seems to follow the one we currently have with the flu vaccine. In terms of immunity with natural infection versus immunity from the vaccine, there have not been enough studies to draw definitive conclusions. The vaccine immunity is based on a single component of the virus, namely the spike protein, and the premise that this protein would be conserved as the virus goes through its cycles of mutation. As I've shown you in the previous diagrams, people that had the actual COVID-19 infection after exposure to the SARS-CoV-2 virus also developed immune memory to approximately 25 or so other proteins found in the virus. And this includes the M and N proteins and other sites like NSP3, NSP4, ORF3A, and ORF8. In theory, this would be a more complete immune memory response to multiple potential targets for antibodies and killer T cells to target, and may increase the chance of an effective immune response to re-exposure to the virus. Of course, even young people can have long-lasting negative health effects from an actual COVID-19 infection, so the added immune memory may come at a significant cost. At the present time, those that have been known to have COVID-19 within the past 4 to 6 months will likely be asked to wait until non-infected people have been vaccinated first. There are always questions about safety. So far, during the phase 3 trials of these vaccines, over 40,000 people have been studied. In the Pfizer vaccine study, less than 0.5% had serious adverse events reported in both the vaccine and the placebo group. 4 cases of Bell's palsy were reported in patients who received the vaccine, while none has been reported in those who got the placebo. Study authors, however, noted that those 4 cases are consistent with the rate of Bell's palsy in the general population. They concluded that there was no clear evidence that Bell's palsy was caused by the vaccine. The most common reaction to the vaccine was pain at the injection site. 63% reported fatigue, while 55% and 38% of participants reported headache and muscle pain, respectively. Less people reported chills, joint pain, and fever after the vaccination, while more people have some reactions after the second dose. There were reports of a severe anaphylactoid reaction requiring the use of an EpiPen in 2 healthcare workers in the UK on the first day of their vaccination rollout. These 2 healthcare workers were known to have multiple allergies and fortunately always carried an EpiPen around anyway. Because of these cases, vaccines were no longer given to people with allergies until more studies are done. At least 2 people vaccinated in Alaska have severe reactions, with one having a septic shock picture response requiring significant medications and medical support for the blood pressure and a 1-day stay at the hospital for stabilization. These patients were not known to have previous allergies, and there is not a definitive reason why these reactions occurred. Likewise, 4 workers in a suburb in Chicago had severe vaccination reactions serious enough that they halted their vaccination program for about a day while the facility reviewed their vaccination protocols. Again, definitive reasons have not been known or disclosed. So at this point, I've answered a lot of the more common questions, but as you can see, there are a lot more unanswered questions at this time. I didn't mention this much before, but we have no meaningful studies on our pediatric population. There is even controversy on the Pfizer-BioNTech vaccine for people aged 16 and 17, as the phase 3 trial data that was released to the public were mostly from consenting adults over the age of 18, yet the FDA granted 16 and 17-year-old emergency use authorization as well for the Pfizer and BioNTech vaccine. It is possible that they have more data than the rest of us have at this time. In addition, we are just at the beginning of this vaccine rollout, and even though over 1 million people are expected to have had the vaccines before the end of the year, many will not have access. There are also surveys that show up to half of the U.S. population is hesitant on getting a vaccine, and at least 20% who would refuse a vaccine even if they were offered one. This leaves the majority of us with the only option of following CDC guidelines, optimizing our immune health, and building up our immune resiliency, at least for the time being. Vitamin D, D3, K2 drops may play a key role, and I've discussed this extensively in my articles and blogs. I've also mentioned zinc support, allergy and immune support, Immunomax, immune defense, Virex, liposomal glutathione, and liposomal vitamin C. All these natural supplements support the immune system and are pharmaceutical grade, properly dosed, and well absorbed. You can click on the links provided, go to the blogs on immune optimization or immune boosting supplements, or contact us by emailing info@jeffreymarkmd.com or calling 925-736-9828. So, I hope you have an intimate, meaningful, and healthy holiday with your loved ones at your home. So take care and stay healthy.